You ever wonder how a tiny little seed can grow into a massive tree? Or how a cut on your finger just heals itself? It's not magic, not at all. It's actually an incredible microscopic dance that's happening inside of you, inside of every living thing, right this second. Today, we're going to pull back the curtain on the cell cycle, the amazing, mind-boggling process that allows life to make a perfect copy of itself. Let's dive right in. Think about it. It seems impossible, right? You snap off a small piece of a plant, stick it in some soil, and poof, a whole new plant. How does that even work? Well, the secret is this incredible ability that cells have to create perfect copies of themselves. And this isn't just for plants. It's the exact same process that lets you grow and heals your body. And it all boils down to one absolutely fundamental process: cell division. All right, so to make a new plant, you gotta have the blueprint, don't you? It's the same for a cell. Before it can even think about dividing, it has to make a perfect, flawless copy of its entire instruction manual. You know, the genetic code that holds every single detail for building and running the whole organism. So, what is this blueprint exactly? That instruction manual is called the genome. It's the complete set of DNA for any organism. And for us humans, that's a staggering 3 billion letters of genetic code packed into pretty much every single one of our cells. I want you to just imagine being asked to copy a 3 billion letter book without a single typo. That is the incredible challenge our cells face every single day. Okay, and this is where it gets really wild. If you were to take the DNA from just one of your cells and stretch it all out, end to end, it would be about 2 meters long. That's taller than most people. So how on earth does a cell pack 2 meters of anything into a space so tiny you can't even see it? And then, how does it copy it perfectly without the whole thing becoming a tangled-up disaster? It's an absolutely incredible feat of biological engineering. Well, the cell's solution to this problem is just brilliant. It's kind of like taking a ridiculously long piece of thread and carefully winding it onto a whole series of spools to keep it neat, organized, and manageable. This amazing packing job is the absolute key to handling all that DNA. So let's see how it works. Okay, this is the cell's masterclass in organization. So it starts with that famous DNA double helix. Then that helix gets wrapped around these proteins called histones. It looks kind of like beads on a string. But it doesn't stop there. This string of beads then gets coiled up even tighter into something called chromatin. And then, for the grand finale, when it's go time for division, all of that condenses down into that iconic X shape we all know as a chromosome. It's an unbelievable journey from a long, messy thread to a perfectly packed bundle of information. And here it is, the final package, all wrapped up and ready to go. This classic X shape is a chromosome that has already been copied. You see, each half of the X is an identical copy, and we call them sister chromatids. They're joined right there in the middle, kind of like dance partners, just waiting for the music to start so they can separate and go their own ways. So, the blueprint is copied and neatly packaged. Awesome. But how does the cell know when to copy it and when to divide? There is zero room for error here. The whole thing is managed by a highly regulated, incredibly precise sequence of events that we call the cell cycle. So take a look at this. This diagram is basically the entire life story of a cell. Most of its time, a huge chunk of it, is spent in this long period here called interphase. You can think of this as the everyday life of the cell. It's growing, it's doing its job, and it's getting ready for the big event. Then comes the main act, the mitotic phase. It's a short but very dramatic burst of activity where the cell finally divides in two. Let's zoom in on interphase for a second. This is the cell's prep time, and it's split into 3 parts. First, in G1, the cell just grows. It gets bigger and duplicates all its internal machinery, its organelles. Then comes the most important part of all, the S phase. S stands for synthesis. This is where the magic happens. All 3 billion letters of that DNA get copied. Finally, in G2, the cell does one last quality check, making sure everything is perfect and ready for the big divide. Okay, prep work is done. The DNA has been copied. It's perfectly packaged. Now, it is time for the main event, the mitotic phase, where one cell is finally going to become 2. Now, here's a crucial point. Mitosis is specifically the division of the nucleus and its precious cargo, those chromosomes. The entire goal, the whole point of this intricate dance, is to make sure each new cell gets one complete, absolutely perfect copy of the genome. And this isn't some chaotic explosion, no way. It's a carefully choreographed performance with 4 main acts, which you can see right here: prophase, metaphase, anaphase, and telophase. Let's walk through each step. So in the first 2 acts, prophase and metaphase, it's all about organization. The packaged chromosomes become visible, and then these special fibers start pulling them into a perfect single-file line right down the middle of the equator of the cell. It's like lining everything up perfectly on moving day before you start loading the truck. Next up is anaphase, and this is where things get really dynamic. Those sister chromatids, the identical copies, are literally pulled apart and dragged to opposite ends of the cell. Then, in the final act, telophase, the cell starts to rebuild, forming a brand new nucleus around each of the 2 complete sets of chromosomes. So, the genetic material has been perfectly divided. But we're not done yet. We still have one big cell that has 2 nuclei. The grand finale is a process called cytokinesis. This is the actual physical splitting of the cell's body, the cytoplasm, where it basically pinches in the middle until it separates into 2 brand new identical daughter cells. And what's the result of all that incredible work? This simple table is the punchline. A parent human cell starts with 46 chromosomes. After this entire elaborate cycle, you end up with 2 daughter cells that are genetically identical to the original, each with its own perfect set of 46 chromosomes. Mission accomplished. Okay, so that's the how. It's a beautiful, intricate, almost unbelievable process. But now let's zoom back out and talk about the why. Why is this tiny, microscopic dance so unbelievably fundamental to life as we know it? Well, this process is the reason we can grow from a single fertilized egg into a complex person. It's how your body replaces millions of cells every single day, and how it heals wounds when you get a cut. And yeah, it brings us right back to our original question. It's how that plant cutting can regenerate an entire root system and grow into a new plant, a perfect clone of its parent. You know, this quote just nails it. It perfectly captures the whole point of the cell cycle. The entire reason for this incredibly complex system is to ensure one thing: fidelity. It is all about passing on a complete and perfect set of instructions from one generation of cells to the next, with no mistakes allowed. The cell cycle is truly one of the most reliable processes in all of nature. It's got multiple checkpoints built in just to prevent errors. But it's not foolproof, which leaves us with one final and really crucial question. What happens when this elegant, highly regulated system breaks down? What happens when a cell just starts dividing and dividing, and it won't listen to any of the stop signals? Well, that's a story for another time.